we've been a bit remiss of not taking questions from the audience, so we'll have some opportunity to basically do both questions here and questions in the panel. And with that, it's time to start the next session, which is health and quality of life. I'd like to ask our moderator, Thomas Goetz, the deputy editor of Wired magazine. Thank you.
So thanks. Good morning. It's fun for me to be back. A couple of years ago, I stepped to the side of journalism for a little bit and got my MPH at the School of Public Health here. And what was apparent to me then was this paradox that technology has created with health. We really have seen technology be a boon for health over the past century, from the vaccines, antibiotics, to the X-ray and MRI. These technologies have really transformed our health along all the metrics that we measure: quality of life, mortality, lifespan. But technology has also become a burden for healthcare. It is often cited as the single largest driver of the increasing cost of healthcare, which, as we all know, has hit 16% of our GDP, $2 trillion, on its way towards $4 trillion and 20% of the GDP in 4 to 5 years. So the question really is, and I think we're going to get at this today with both from the healthcare side as well as tapping the venture capitalists and engineering experts we have on the panel, is how can we deploy technology to really improve public health and individuals' health so that more people have better health at cheaper costs. So with that, I wanted to start with, and one thing to note, we have, over the next hour and a half, we have 5 people we're going to cram into an hour and a half, so it's going to be tight. Jay Keasling, who's going to talk about synthetic biology, will have a format similar to the other ones, but then we're going to kind of break up into a bit of a 2-for-1 thing that goes on, and hopefully it'll work well. But anyway, let me bring up Jay Keasling.
Thanks very much. It's a pleasure to be here. I want to throw out some challenges, and these link some of the work we were talking about before and some of the challenges with some that we'll be talking about in the next few talks. The first is nitrogen fixing crops. We spend about $3 billion every year for about 12 million tons of fertilizer in the U.S. alone to fertilize basically corn and wheat. 1 ton of fertilizer consumes about 33,000 cubic feet of methane. This is 1/3 of all energy used in the U.S. agriculture sector and 1% of the world's total energy consumed on fertilizers. Let me throw out another challenge. We use 14 million barrels of oil every day. And there are now significant mandates for supplementing or replacing that petroleum-based fuel with biofuels. And yet, we don't have an energy crop. Corn, for instance, requires significant amounts of water and fertilizer. And the fuels that we're producing can't be used in our traditional infrastructure, and particularly in diesel engines and jet engines. We need the development of dedicated energy crops with reduced water and fertilizer requirements, and microbes engineered to produce advanced biofuels that can be used in our existing transportation infrastructure. And a final challenge, specialty and commodity chemicals. Worldwide, this is a $1 trillion business. It's multi-billion dollars in the U.S. alone. These are things like polymers. Basically, everything you're interacting with in this room comes from petroleum in some form. The coating that's on the wood floor, the material that in this fabric up here, everything that we're interacting with comes from petroleum practically. And yet we're running out of petroleum. So how are we going to replace that? And a final challenge that'll link in with healthcare. Can we develop novel therapies? For instance, a tumor-seeking bacterium that you could inject into your body that would hone to a tumor, would inject that tumor with a drug, kill the tumor, and then kill itself. Okay, significant challenges. These are only reachable, I would pose, through industrialization of biology. And this is different from genetic engineering that we have been doing today. We call this synthetic biology. And it's really the construction of reusable, standardized biological components that can be used in combination to solve many particular problems. Let me take a couple of industries, and then I'll contrast that with how we do biotechnology right now. Let's take this industry that many up here know so well, the microelectronics industry. We have computers that are so inexpensive and yet so powerful because of an industry that was created for developing parts that could be combined and used in combination to build these devices. If you have a part in your computer that goes awry, you can go to a place like Radio Shack and pick up a new hard drive, for instance, and put it into your computer. And if you're a maker of those components, you can sell them to the makers of boxes. So Dell, Toshiba, Lenovo, Apple, they don't make the components in those boxes. They add them together and make them palatable to the consumer. This works because we have things like standardized ports on those components, like the SCSI port, for instance. You don't have to worry about that hard drive not fitting in your computer as long as you have the right port on the back. And of course, we have models that allow us to design components like the silicon wafer, and this is actually something that Richard Newton was very much a part of and helped pioneer. Let's take another industry. This is the chemicals industry, and I have here one of the largest bulk chemicals used, styrene. It's used in things like toys and styrofoam, and a lot in our tires, actually. If you wanted to produce styrene, you'd build a chemical factory to do so, and you'd pull together some not so off-the-shelf components, but still things we call unit operations, things like reactors and distillation columns, and you'd connect them with pipes. And these are all standardized by the chemical engineering community. Of course, we have standard connections for those. If you think about going out and buying a pipe at a hardware store, you never have to worry about the manufacturer that those pipes came from. All you have to worry about is that you got the size right, because somebody took the time to standardize the threads on those pipes. Now, what these two industries have in common are parts and devices that function independently and are well characterized. We have standards for the connections on those components. We have standards for how we characterize those components. And when we need something new, we can write models, design, and fabricate those components. Let's take a different industry. Let's take the biotechnology industry, and I want to target a project that we've been working on in my laboratory, and that is an anti-malarial drug. How would you develop an anti-malarial drug that would treat 500 million people, that would be available to them and affordable, and reduce the lives lost, nearly 1 million and counting each year, due to malaria? Well, there's a drug out there. It's called artemisinin. It's an ancient Chinese therapy. It goes back to about 150 BC. It comes from a plant called Artemisia annua, and it's too expensive for people in the developing world to afford. So we asked the question a few years ago, can we engineer a microbe to produce artemisinin, stabilize the world's supply of artemisinin, and at the same time decrease the costs? And the answer is, yes, we can. It's change you can believe in. We decided that we wanted to synthesize this drug in a microbe, but do we have off-the-shelf parts? No, we don't actually have off-the-shelf parts. There's no BioShack out there where we can go pull those parts off the shelf and combine them together. We actually have to go into the organisms. In this case, we went into yeast and into the plant Artemisia annua that naturally produces this drug, and we had to pull those parts out. But the problem is, because we have no off-the-shelf parts, we have no standards, no models for how to combine them, and no connections to fit them together, you run into problems like intermediates in the metabolic pathway accumulating and killing the microbe that it's supposed to produce this drug. But with many years of work, in fact, about 6 or 7 years of work, we've come up over 10 million-fold in the production of this drug, but at a significant cost. It was about 150 person-years worth of work, and at a cost in terms of the science of about $25 million. Fortunately, we finished the project, we finished the science. It's now at a pharmaceutical company where they're scaling up the process, and we hope that by 2010 we have the drug out for people in the developing world at a reduced cost, stabilizing the supply. The goals of synthetic biology are to reduce those incredible times that it takes to develop something as simple as a microbe to produce this drug. We want to reduce the cost and the time to engineer these biological systems. And at the same time, we can use this technology to better understand how biological systems function by reconstructing them. So here's a roadmap for synthetic biology. And this is actually a roadmap that Rich Newton worked on in his final days, developing an educational program to actually train a new generation of bioengineers, a research program to determine how to set the standards on those biological components, to learn how to develop standard components, and then to develop a biofab, like the fabs we have for the microelectronics industry, to develop bioparts, biocomponents that we can offer open source so that companies aren't patenting the parts that they are developing, but they're patenting the applications that use those components so that those components can be readily shared and so that we can further develop a new biotechnology industry. And then, of course, the applications. And I want to just mention SynBerc, the Synthetic Biology Engineering Research Center. This is a group of universities, MIT, Harvard, University of California, San Francisco, University of California, Berkeley, Prairie View A&M University, who've come together, brought all the scientists, or many scientists who work in this area of synthetic biology, to work on many of these problems. And this is something that Rich Newton helped us develop and actually was a huge proponent of. So here are some of the applications, and I want to just throw these out as stretch goals for the area of synthetic biology, because I don't think we're ever going to achieve these goals if we don't have synthetic biology. When they had the first Asilomar conferences in the early 70s, after genetic engineering had been developed, one of the goals was to develop crops that would be self-fertilizing. It's been 35 years, and we still haven't developed those crops. We need to develop those crops to save energy. They should also save water, because there's a huge need for water if we're going to develop energy crops. And we need to increase their photosynthetic efficiency. Plants have actually surprisingly low photosynthetic efficiency, around 1% to 3%. We need to at least double that. I wish Shai were here for the second point. There are no batteries, and probably not going to be batteries for jet engines. We need to develop, therefore, biofuels that can go into jet engines and into diesel engines. And these have to be no-compromise biofuels, biofuels that will fit into our existing multi-trillion-dollar transportation infrastructure. And I think this is going to be a significant challenge, but one we're up to. And finally, nature has given us a huge number of molecules, and nearly 60% of the drugs we take now are based on these molecules. And yet, almost every pharmaceutical company that I know has dropped their natural products discovery groups in favor of combinatorial chemistry. And this has led to a huge drop in the discovery of the number of drugs and drugs out there that are available. I would propose that we need to develop all the chemistry that exists in every plant and every strange microbe on the planet and put it into a microbe or two that could produce every natural product available, and therefore increase the number of drugs coming into our drug pipeline. Because after all, evolution has done wonders for discovery of these molecules. We need to use that. And finally, I'd like to just thank this man for helping us to develop synthetic biology here at Berkeley. He was a tremendous influence on me personally and has been a tremendous influence on the development of this field.
So synthetic biology, I think, is certainly one of the more provocative disciplines that has emerged over the last 10, 20 years. And I think one of the things that you mentioned was the roadmap. And I think that's a rather unique approach that the synthetic biology community took at the outset, at the birth of the discipline, to really think about structuring the science and making sure that it had the potential intersections with industry at every step of the way. And I'm wondering kind of if you could just elaborate on how innovative and kind of what different approach that took to make sure that that was an aspect of the way you planned.
So I think the one thing is that we've tried to learn from other industries. We recognize that the biology industry or a new biology industry is not going to look like exactly like the chemicals industry or the microelectronics industry or any other engineering discipline out there. It's going to be its own unique discipline, but it is going to share or should share some of the best aspects of these other engineering disciplines. This idea that you can design and build de novo, that we should have scalable factories for building some of these components so that users, people who are trying to solve some of these challenge problems, have a source of tools to build their particular solution to that problem. So we've tried to learn specifically from these other disciplines. Now, there's one point I think that's really important that has been very unique about the synthetic biology community and the synthetic biology field, and that maybe these other areas haven't had to worry about, and that is, so you develop these components, and they're easy to use, and any kid can now order them and start to do biology in the garage. And gosh, maybe you can order a Goetz cell, and you can order all the components to build a chromosome, and so now you've got a new bacterium that you can build in your garage that your children can build. What are the societal implications if you can do that? And there are some significant societal implications. And one thing I think that's very unique about the synthetic biology community, and maybe this is a self-preserving role that we've taken on, is that we've tried to think through these societal implications. How should we regulate biology, the industrialization of biology? Should we give everyone a license or license people to be biological engineers so that you can only order those components? Should we regulate the synthesis of DNA in some way? So I think the synthetic biology community, maybe more than any other community, has tried to think through these ahead of time. I can't say that we have all the answers, but I think in some ways we've been unique in that way.
Jay, in the execution or the creation and the execution of the semiconductor roadmap, one of the big exercises is always to identify the roadblocks, the big things that we have to address from a scientific perspective or a scientific community perspective. So what do you see as the biggest roadblocks for synthetic biology, let's say in the next 5 years, or something that have to be addressed first?
Only one. I think there are many roadblocks. One is that we have to recognize that biology isn't completely known, and some might say it's rather poorly known at this point. And I've had a lot of arguments of how can you possibly engineer biology, and I would argue that microelectronics, so we didn't know everything about physics, but the microelectronics industry developed and many other industries developed. So I think first we have to recognize that, but I think there are things we can do, and one of the reasons I, as an engineer, work with microbes is because I'm able to engineer them and do a few things that actually do some engineering on them. So some of the challenges I see that I would really like to have, I'd like to have standards for some of the biological components we use. There are no standards for these, and it makes them very difficult to recombine and reuse. And we haven't seen a lot of that in biology. In many parts of molecular biology, you generally build something to be used once and not recycled. That's changing, but slowly. So I'd like to see standards. I'd also like to see a parts fab, a place I can call out and get a promoter or a ribosome binding site. And it doesn't have to be perfectly tuned, but it's got to be partway there. And that would be much better than what I have now. And that is, every time I want to use a new component, I've got to completely retune it and study it in detail. And this means that the development times and therefore the costs are high. So if I had to think about those, in my mind, would be some of the greatest hurdles I'd see us jumping over to make this field work better.
Have you had conversations about this with Gates and Path and some of the other organizations? Because it seems to me, putting this in the context of the millennial goals and the UN strategies and showing how this would really broaden the effectiveness of investment in invention in this area would be a terrific way to frame it.
Right. So the malaria project that I talked about was actually funded by the Bill and Melinda Gates Foundation. And so I would say that in many ways they're very well informed about this. But we also have to recognize that artemisinin is unique in many ways in that it's a drug that's known, that works, that's supply limited and too expensive. And there aren't that many drugs like that in the world. So, but what I'm suggesting is that we can use this as a mechanism, synthetic biology, to accelerate both the production and maybe the discovery of drugs.
Can I ask the panel, so especially with vaccines, there's been a notorious market failure over the decades in terms of creating viable vaccines that will be produced. And I'm wondering if anybody has any analogies in terms of the market failure things that synthetic biology might be able to accomplish.
Well, at a meeting we were just at, Jay and I, the people were talking about to try and attack HIV, instead of using a vaccine approach, perhaps you can use a bacterium or a microorganism which is in the infected areas, which is in a commensal bacterium of the humans, which are secreting peptides that would block the infection process. So there's ways in which one can begin to think of the synthetic biology approach to making alternative to standard vaccines.
So the first major barrier, well, there's a couple of them. One is money, because everybody says it won't work, and that's how you get up higher and higher, at least in my experience, in the pharmaceutical organizations, because you save money right away. The second is what you hinted at. We really don't know enough biology to say we won't do harm at the beginning. The dream has always been there to be able to treat infected HIV people with the vaccine, and we can't yet prevent uninfected people from getting it because right now the virus is still smarter than us. But we may catch up. But when we introduce treatments to try to vaccinate against cancer, and I'll talk about that later, it's in a field of biology where, although we've done a century of work on it, it's just now accelerating to the point that we start to understand the principles. So I'm not saying that it's going to be impossible, because if you talk to an astrophysicist, he says the minimum amount of time I study is 10,000 years. And of course, in 10,000 years, and well before it, synthetic biology will probably take over the manufacture of a lot of these things. But we have to recognize money, the FDA, the do no harm, are really major problems in any of these ideas.
I want to be sure these two microphones that are in the audience that have been cold today might get a little use. So please come up. We have a couple more minutes on synthetic biology. I was wondering, while people are making their way to the microphones, I was wondering also about you showed a slide of BioShack, but you really are actually well along the way towards creating that kind of armada of tools. And I just wanted to kind of get you to elaborate on, I know BioBricks and Codon and some of the other synthetic biology groups are working on exactly that problem.
That's right. So I think one thing that's changed in the last few years is the startup of many of these DNA synthesis companies that will do long DNA synthesis. And this has been tremendous for those of us who engineer microbes to do various things, not even necessarily microbes, but just I think the biotech community. I think this has been very helpful. And now the costs are getting down where just about anybody can, with research grants, can go out and order some genes. And you can order just about anything you want as well, which some of these companies are trying to address. There are also efforts to try to put components that are characterized up on the web so that people, if they have the sequence and they have the characteristics for it, can send it out to a company and have it synthesized. One of the examples is a registry of standard biological parts that has been developed at MIT for serving both the sequence of the particular DNA part and its characteristics as input-output characteristics. And again, with the sequence and with those characteristics, you could just go out and synthesize them. Now again, they've got to be standardized. You've got to have standardized measurements, and so that's being worked on as well. But it's a very young field, and we need development. But given kind of the strength of the biotechnology area, I certainly think it has huge potential.
Well, yeah, it certainly is exciting. So I want to move on to the next session, or the next clump of speakers. And what we're going to do is we're going to have two people address kind of two sides of the cost-effective and cost-effective coin, if you will. First, we're going to have Paul Camuti from Siemens, and then... Oh, other way around?
We're going to do it the other way.
Okay, so first we're going to have Reg Kelly from QB3, and then we're going to have Paul Camuti. But what we're going to do is they're going to each talk for 7, 8, or 9 minutes, and then we're going to flip right to the next speaker, and then we're going to go towards the bigger group. So if you guys will bear with me on that, that should work easily. So Reg, thanks.
So thanks very much for being here today. It's a great honor to be at an event dedicated to Richard Newton. So I'm going to talk about health and the quality of life. I'm director of QB3, which is a consortium, a rather remarkable, unique consortium of Berkeley, UCSF, and UC Santa Cruz. We address synthetic biology, but that's already been talked about. We also address health issues, and we do the standard things. You know, we do drugs, diagnostics, and devices, and we address most of the major diseases. So the challenge for today was to think, well, what is really, the moonshot, right? What is something that's really special? Is it some great new breakthrough in Alzheimer's disease, or should we be looking for something in malaria? What is the thing which is probably the most exciting to me? So the thing that was actually turned out that I really wanted to address today, if I can, is in fact cost. What is really the thing which is going to, I think, going to have the biggest impact on the health of the planet is to reduce the cost of healthcare. Now, you've heard already some of the problems here. In the international level, if you look at how much things cost, 1/3 of the population is spending greater than 10% of their budgets on healthcare. That's one of the major reasons, for example, in China, that the poor people out in the rural areas are having to sell their houses because they have to try and generate money to pay for healthcare for their families. So it's a huge problem, and it's completely asymmetric, where 22% of healthcare budget goes to 1% of the population. If you look at the national level, as was alluded to earlier, we have a huge amount of our budget being spent, greater than $2 trillion per year, $6,500 per person. That's twice what is spent by other major nations, with no increase in life expectancy. So there's clearly something wrong with the American system economically. But it's actually even become more local. Even though we spend so much nationally on health, we also, even within the state of California, spend a huge amount on health. So in fact, again, within the state, the cost of healthcare is going up 8% per year, whereas the consumer price index goes up about 4%. So there's huge problems with paying for healthcare. Now, there's also a large library of books about how to deal with these problems. And I've looked through some of them, not exhaustively, but there's a whole range of things which are suggested. And I'm not going to go through them all. You've probably heard most of them. But what is singularly absent from each of the books we're looking at is to try and see, can you actually develop technologies, which are the sort of things that is our bread and butter, which could in fact bring down the cost of healthcare. Now, what is immediate? Why is there an absence of these technologies? Well, it goes against the average market model, right? If you went to your board and you said, "I've got this great idea for coming up with a gizmo that reduces the cost of healthcare," you would be laughed out of the room, right? That's not the way economics of medical care works. So there has to be some other system. So how could we think of a different system? So this sort of slide just sort of shows something. These are just entirely qualitative. It gives you the feel for what the problem is. You know, as we get sicker and we move from being healthy through home care and ICU, the quality of our life singularly diminishes, right? Especially when you get an intensive care unit. But at the same time, cost goes up dramatically. So the huge cost in medicine is large part of it is in the intensive care units, right, and in hospital care. So if we want to impact the economy here, what we have to do is intervene early, find ways of keeping people healthy and stopping them getting into the intensive care unit. So how might you do that? So let me just give you an example of one or two of the sort of technologies that are going on in QB3, although there are many of this type. So one of the things, dealing with pathogens, somebody comes into your emergency room with a fever, you don't know what is making them sick. Is it a virus, is it a bacterium, what type it is? So you go through the list of suspects, and that can take a long time. A much better technology is being developed by Don Gannam and Joe DeRisi at UCSF, where they're making a viral chip which diagnoses every virus that's known to man, whether that's a human, plant, or animal virus. They can recognize it, and so they can tell in a bodily tissue what virus is there. So how can you use that? The way they're using it is actually quite intriguing, the way they're exploring right now. How can you monitor the average health of a city like San Francisco? Well, what they're doing is they're looking in the raw sewage, right? They're monitoring the raw sewage to see what viruses are appearing, with the idea that this is a way of getting a quick alert of what might be happening in a city. So when somebody comes into an emergency room, the physician has been warned what is more likely to be the cause of the disease, right? It's a way of keeping down the cost and keeping people healthy. There's similar technologies for developing bacterial pathogens coming out of Affymetrix. Can you diagnose cancer early? Well, there's techniques coming up, both proteomic and genomic, in order to get early diagnosis of cancer, possibly from blood. But the problem here in the techniques that are currently being used, like in genomic health, is their tests are too expensive. If you're wanting to have routine testing of people every year, you've got to keep the cost down in a couple of $100, or you're not going to be covered. So how can you keep down the cost of these complicated tests? Well, the sort of technology, and this is one from Richard Matthews, who's at Berkeley here, is to try and use microfluidics. Microfluidics is great. It takes advantage of the strength of the Bay Area, because it's essentially the same silicon etching technology as one uses for chips. But you can actually make a device like this one here it's a PCR machine, which can do a PCR reading in 10 minutes instead of the hours that are normally taken. How does that save money? Several ways. One, you use tiny amounts of reagents because it's all in nanoliter quantity, right? Secondly, all of this, these heaters and built-in things allows the chip to do the whole procedure. You just put the sample in, the chip does all the analysis and actually sends out the signal giving you the measure of what the diagnostic is. You don't need expensive technicians. It's expensive technicians that really push up the cost of diagnosis. So by having the logic on a chip, you can do it. Also, if you make them cheap enough, they're disposable. That saves a whole lot of problems in this way. So this is another type of technology one can develop. What about in the area of pharmaceuticals, getting drugs? Well, we all know it costs about an average $1 billion or so to bring a drug to market and takes about 13 or 14 years. Something has got to be changed about that. So what is getting cheaper in our lives? Well, what is getting cheaper is computation. So what we're developing here in QB3 are ways in which you can use the power of the computer to predict drugs. And the key thing is a drug will fit into a slot on a protein and inhibit a proper molecule from getting into that place. You can either do it by screening through millions of compounds, which is a standard thing that Pfizer and Exelixis do, or one actually do it computationally. You say, this is the shape of the drug, this is the shape of the groove, let me computationally fit one that works. That's working remarkably well now. So for example, this is a protein which has a drug fitting into a groove on it. This is actually one developed for Chagas disease. But what one is able to do now in a paper which was in Nature is you're actually able to predict, take a protein that you don't even know what it does, and calculate what sort of molecule must fit into the groove, and then predict what that protein does. So this ability to predict what small molecules fit with a target is developing very, very fast. And some really exciting ways we're developing to take drugs which have already been FDA approved, right? So all the work has been done, and repurpose them for functions which hadn't been recognized before. We just use the computer to search for new targets, and therefore do things much more cheaply. So the final thing is, and this is very unfortunate to admit, is that much of the surgery that we do at UCSF is unnecessary, right? So particularly true in breast cancer and prostate cancer. So let's look at the prostate cancer. You know, it's the major, 18% of men have it, 3% of men will die from it. It's one of the major causes of death after cancer after melanoma. And so many of the surgeries are unnecessary just to be safe, right? And probably about 90%. But surgery has its negative sequelae, right? You can get incontinence and impotence at about the 20% level. So if you can avoid surgery, it's a big advantage. Why do we do it so frequently? Because we can't tell whether the cancer is metastatic or not. So we could get ways of telling whether this is a metastatic cancer or we should just let the patient go. This would be a tremendous improvement in quality of life and a tremendous savings economically. So a technology that we're developing in QB3 is to take use of a C13 compound, which is non-radioactive, but you can hyperpolarize it, and you put up its spin signal about 50,000-fold, quite a remarkable improvement for anything biological. So what you can do with this drug, with this chemical, is measure the conversion of pyruvate to lactate. Why is that important? Because a metastatic tumor, due to a Warburg's effect, which has been known for decades, it has a hypoxic and it has a different metabolic rate. So you can actually tell a tumor is metastatic by looking at the rate of conversion of pyruvate to lactate. So the technology that's being developed in QB3 to do this will allow us, hopefully, to actually discriminate and save 90% of the unnecessary surgeries in here. So let me end by what is the moonshot? What I'd like to do is propose that in California, and we should start things here because this is where so many things start, we should create an institute for cost-effective health technology development. That what this institute should do is first go and say, what's the low-hanging fruit? Which one of those diseases are most, could we save most money on if we were to do something? Then we decide which one of those could make a lot of money, which ones do we have a technology to address, and then we go out and put an RFP out for those technologies to the incredibly creative people we have in California. Now, of course, the problem is, how do you pay for all of this? And so one of the things, maybe the thought is, maybe we can make it like whistleblowers. You know, whistleblowers get a little fraction of the money that they save when they save the state money. Perhaps we can do something like this. This institute will take back 20%, whatever the state saves in bringing in the technology it introduced. You know, if we had an agreement like that, we could probably raise money to get the institute going. So what are the side benefits of this? Partly, I think this would benefit because developing these technologies could be the beginning of a new market for cost-effective healthcare technologies, which we could sell around the world. And finally, one could begin to think using this technology to help the other places in the world, which are outside California, which have needs which are even greater than ours. So let me end there by saying this idea is not completely crazy. I actually got a politician in California to put it in the state budget this year. So this was actually, QB3 was funded to do this work, but you can imagine what happened when it went to the state legislature this year. It's cut. But at least the idea has got some sense, and I think we have to continue to develop it. Thank you.
Hi, I'm Paul Camuti, and I'm with Siemens Corporate Research. It's really an honor for me to be here. Richard Newton, I know, has a connection to just about everybody that's here and was a big part of what we've done in our company as well as what we've done here in California. So I think we're going to have a good transition, Reg. That's the good news. I've also learned a couple things. When Tom mentioned earlier about population growth, he said, however that started. Jay, I think that's kind of the question we need to answer with synthetic biology, because that might be a barrier to moving some things along as well. And also, I'm not too worried about the bacteria and the science kits, because when you buy chemistry kits today for our kids, there's no chemicals in them due to the regulation that we have there. So it's good to know that we're addressing kind of the diffusion of the technology as it goes in. What I wanted just to spend a couple of minutes on was kind of how the technology related to biology, genomics, proteomics can fundamentally impact healthcare, and that's something that we're working on. The context of this is actually in our company, but this is actually at a much larger scale than any one of the industry players could do. As you know already, the way that we deliver healthcare today, it might require some grounding actually relative to the science, is that anybody who has to interact with our healthcare system, you might have a best case scenario actually in some of the facilities here in the Bay Area. But generally we're practicing not science and art, and it's a pretty reactive delivery of healthcare where we basically wait for symptoms. We have coarse diagnostics and screening, and we have a tendency of, depending upon who the actual doctor is, doing a one-size-fits-all type of therapeutic. There's a lot of pressure on healthcare costs. There's an incredible and rapid advancement of technology, fundamental technology that can transform the way that we deliver healthcare. For me, it's somewhat, I keep reminding myself that the technologies involved here are actually a little bit fresher than the technologies of IT. And so we really are just at the beginning stages of technical discovery to impact the delivery of healthcare. And of course, in some circumstances, we'd actually like some choice over how we drive healthcare, which is generally not done. And so, the great vision, the direction of the future is how do we make healthcare more personalized, where it can be more predictive, preventative, and tailored to the individual disease states. You've already heard a number of different things. Of course, we have a tendency to take kind of an engineering perspective to process improvement, huge expenditure in the United States and a growing expenditure globally. And we have to bring these technologies into specific focus in order to be able to knock costs out. And in the area of personalization of healthcare, it's being able to do diagnostic tests, both in vitro and in vivo diagnostics, in order to get a stratification of patient populations so that we can tailor the therapies to the specific patients. You might be aware, we could bracket the opportunity for cost savings in a number of different ways. One statistic, just in terms of adverse drug reactions, about $100 billion of healthcare related directly to either misprescribed, overprescribed, or ineffective therapies. One great example is warfarin. It's a blood thinner. It's typically, if you're in a situation where you need to have that, it's prescribed based on a bunch of traits, how tall you are, what body style you have, what your weight is, and more than 50% of the time it's not right in the original dosages that are given. Recently there's been a genetic marker test for that's greatly improved the ability to dose warfarin, and it's reducing significantly the amount of adverse reactions and the amount of time that it takes for effectivity of that drug. So through the specific application of these technologies, we can get a more direct assessment, and we're able to dramatically improve healthcare. The big problem, and you'll be glad to know I'm not going to go through every block on this chart, is, but just to make the point that the healthcare and diffusion of technology in healthcare is really impinged by the stakeholder management, right? And actually, it would take a few people some time to realize that actually the patient is a stakeholder in the delivery of this. So from the physicians to the payers to the regulators to the government to the pharmaceutical industry and to diagnostic companies like ours. All have competing interests, all are trying to optimize their own individual pieces. And in order for us to diffuse and really dramatically change the way that healthcare is delivered, there's a pretty large stakeholder management issue. So, the general view, and again, I would apologize that this says Siemens, because as I said before, this is an industry challenge, which is how do we bring together the in vitro diagnostics, immunochemistry, molecular information, genetic profiling, proteomics, with advanced imaging systems which allow us to quantify and categorize in the body different disease states to optimize therapy, and to do this by leveraging information technology, right? So the problem, or one of the challenges as we go through any one of the individual technologies that we would apply, we're always looking for kind of the analytical quality of those instruments. But in order to translate this actually to impact on healthcare, we have to define the clinical efficacy of those treatments. And so it really requires a lot bigger collaborative effort in this direction. But the view and the vision of the future is that by combining these diagnostic modalities, that we're able to dramatically reduce the costs of healthcare while improving the quality. Thank you.
So great. Well, I have lots of questions, but I also want to be sure we get as much of the panel in as possible. I guess one of my first questions is kind of looking at the two sides of this coin, and I don't mean to put you on the spot, but one of the things that is often cited as an inflationary pressure in healthcare spending is imaging. And radiology departments are cost centers, and when we look for cost effectiveness in healthcare, technology just doesn't seem to work the way it does in other industries. So I'm wondering about that, kind of why is healthcare different in terms of the way technology gets deployed for improving our costs?
Yeah, so I think one of them is the stakeholder chart that I put on the screen there, is that there's not a really good alignment of those stakeholders. So of course, imaging does get a lot of the focus. But just to give a simple example, there's not a way today, if we have a personalized in vitro diagnostic test that improves the efficacy of a particular therapy, right? That would require somebody has to pay for that, right? So you'd have to have a reimbursement code, be able to go to a hospital and get tested. And what that's going to do is reduce subsequent treatment, right? And so the economic analysis has to link across the whole value chain. And for anyone who knows anything about the way that we get that economic analysis through regulatory, linking together value chain and actually thinking about a system doesn't come into play of approving individual treatment to the extent that it should. We're getting better, right? But this is a major challenge, and so the way that we approve diagnostics is a big part of the problem.
Please, Molly.
That's one of the reasons why I think what Reg proposed is very interesting, because putting the filter of cost effectiveness right up front is something politically we've been very loath to do in the country. It's been considered sort of verboten to say we want to put a cost screen or a cost efficacy screen on healthcare diagnostics or interventions. And when you do that, if you just take the example of imaging that you were giving, we can see that coronary angiography, CAT scanning, which is quite expensive. It's a 64-slice machine. It's a big decision for a lot of non-academic institutions, is probably going to be very cost-effective at the early diagnosis of acute myocardial infarction. So if you have a good cost-effectiveness screen for this, especially in the development stages, when the investors are actually underwriting the development of a technology, you may actually be able to differentiate, and you'll find some of the imaging makes a lot of sense, and a lot of the diagnostics and interventional pieces besides biotechnology. I think one of the pieces we're missing here is the information technology-enabled devices, which is the biggest, or the really largest potential savings in the next decade in healthcare. But it would fit very well with the structure that Reg was laying out. And so I think that the concept here of trying to solve problems and lower cost it would be very workable. And it would justify a lot more investment in public policies that are causing the fragmentation now.
Yeah, please.
Yes, well, thank you for inviting me today. And I'd like to make a comment. I'm listening to these talks about energy conservation and keeping people healthy. And I'd like to introduce a concept of the human energy unit. We'll call it the BU, the brain unit. And what I'd like to argue is as the number of humans has gone up, the value of the BU is not keeping pace. And the reason it's not keeping pace is because of the problem of socioeconomic effects around the world. And it's been clearly shown in several recent studies in the last 4 or 5 years that if you are from a lower socioeconomic background, the key part of your brain that we'd all like to have to be most active in terms of inventiveness and creativity, your frontal lobe, is actually impaired. It doesn't develop properly. And you say, well, how does that matter to engineers? Well, in fact, everything we're doing here, yes, we need biology to keep us, standing up and upright and support everything, but everything that's going on is electromagnetic right now. It's really electrical signals are producing all the behavior that we're using as we listen here and listen to these very interesting talks. And it turns out that those signals are actually now recordable and trainable, which means there is a possibility to introduce engineering concepts into brain neuroscience so that you can take kids, or at the other end of the spectrum, older adults, and improve their ability to handle and process information and be flexible. And those studies are in progress. And what you get then is a smarter BU who makes smarter decisions. You avoid things related to simple things like conduct disorder or bad decisions related to drug use. So I would like to see I'd like this prize for the most energy efficient home. I'd like to see a prize for people who are trying to develop novel ways to improve the BU around the world, because going in and making people healthy is great and stopping malaria is great, but if you really want those economies and those people to go to the next level, they need a better BU.
So that seems to tie together for me in terms of behavior. So now tell me if I got this wrong, but if you want to get people to make more use of their brain, they have to change their behavior. And certainly that has overlaps with healthcare decisions and how we're trying to make, a lot of the cost savings are incumbent on making better decisions so that we don't get into these kind of expensive care paradigms. And does that, is a better BU really translate into better individual choices or better societal choices, or?
It would translate into better both. It's quite clear. You can take, there's a study going on at Berkeley. You take inner city kids from Rosa Park Elementary School, bring them in, put them through physiological tests of how their frontal lobe works. They're not doing as well as higher socioeconomic cohorts. You put them in a 4-month training program and they're better. If I told, I'm not going to point to anybody based on age, but if I told you, if you treat the brain as an organ, it's a complicated organ, it's got multiple sub-organs, and start thinking of it just as not some metaphysical concept. Think about it like your heart or your muscle. Nobody would argue that you couldn't improve muscle function and heart function by training. And that's exactly what you would be, that's the exact idea about the brain training for kids or for adults. If I told you you're going to put a little strap on here, 3 days a week for 30 minutes, you did some interesting mental exercises where you looked at brain activity and tried to integrate it with what you're doing and improve your function. And I told you were going to offset Alzheimer's by 4 years, everybody'd be wearing these things. So it's an engineering problem in the end.
May I comment on this? Because actually my background is in neurobiology. One of the things one has to be a little careful about is that for Alzheimer's, there are 4 different tests of this type which are being used to try and improve brain function commercially. The trouble is trying to measure what do you mean by improvement. And when people were able to come up with quantitative measures of measuring improvement, 1 of the 4 worked, but 3 of them actually had no use at all. So what one really has to do before one brings in any metric of this type or any potential improvement of this type is have some clear metric that you can show quantitatively that there's an improvement, and that's not easy.
Yeah. As a neurologist, and actually a practicing neurologist, I think a lot of these things are going to be better on the preventive side. Once you've got the disease and the plaque burden, you've got a whole other issue in terms of, the horse has left the barn. In that, not to get everybody too nervous about this, you probably know the state of there are 4 studies now around the country, one out of Berkeley, that if you take healthy 70-year-old, healthy, perfect mini mental status, and you do a PET scan on them with this new amyloid tracer, 10 to 14% have amyloid deposition. What does that mean? Are they going to go on to Alzheimer's? Can we stop that? This is a huge potential public health issue for us to be thinking about.
So one of the things I wanted to make sure we addressed was the issue of just the amount of data processing that's, Reg talked about this in terms of the computational power, but a lot of shifting, making better healthcare decisions, both individually and as a society, is making better use of data. And I'm wondering if anybody has any insight into kind of the way that a kind of data-first strategy might help speed some of these cost savings along.
There's a whole lot of very early stage work going on in data mining in healthcare, but it's very uncoordinated and unfocused. And nobody here has brought that up as a major concept to throw onto the table, but I think as an opportunity for academic research working with industry, there's a huge potential here because there are enormous amounts of data collected. I mean, Siemens collects huge amounts of it. Any one of the EMRs do. You get it out of the PACS and the imaging systems. And we will have biologic information at a growing pace, and the ability to integrate that and produce really important discoveries is an exciting possibility, but there isn't much investment in it yet.
I kind of skipped over it in my presentation between the in vitro and in vivo diagnostics and just assume that there's healthcare IT. We're in the middle of a very large EU project to leverage actually the grid infrastructure in the EU to link together 12 clinics for pediatric diagnostics because the specialty diseases in kids don't present themselves in enough quantity in any one place in order to be able to get the data in a fast enough amount of time. But kind of our thinking on this diagnostics question or data analytics question is Moore's law is always interesting. Exponential curves get more and more exciting the longer you hang around. And so we're at a point right now where we're able to put information technology and data analytics to do quantification out of the images that are there to get better correlation between all this data that comes in, because to the earlier point, right, if you can't quantify and connect the clinical relevance of some of these tests, you're not able to get into the cost efficacy of it. So there's 2 real areas in healthcare that IT is important. I think one is this data analytic piece, and the other is to link together, as Molly, you might have indicated before, the health record, because the point of care happens in all different ways at all different times. And so to come together with a consistent IT strategy in the United States to link together health records is also a pretty important piece of it.
So I was on the first commission of the National Academy of Sciences to look at AIDS and HIV in 1985-86. And they wisely included people in the science and the medical side and the insurance companies. And this is the point I'm trying to make. If you don't include the insurance companies or who pays for the care, you find out something very bad, or if you include them. So we came to the point that we understood that there were diagnostics for AIDS. This is 1985, 1986, that we likely knew the agent, we knew the way to go forward. And from a public health point of view, you have an epidemic that's spreading by known means. And the judgment then was that it's better not to have it in the public record who has HIV and who has AIDS. Because as the insurance company executive told us, we will exclude you. And therefore, until you have a reasonable health care payment system where you can't be excluded, the information is dangerous and people will withhold it. On the other side of it, we've had many, and I'll talk briefly soon, advances in therapy which will cost a lot of money in the beginning and then eventually will go down. And when somebody has their venture capital or pharmaceutical company hat on, they say, well, this is going to be too expensive. I don't know what your business model is, blah, 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 blah. And then when their wife gets breast cancer, they call me and they say, what happened to that?
Well, unless anybody has anything else, I think we should probably go to the next session, which is on cancer and innovations in cancer research. So I believe we're going to start with Frank Douglas, right? Okay, and then again, we're going to do one of these quick turn things. Frank is going to talk for a few minutes, and then we're going to go to Dr. Weissman. And those microphones must be used before the end of the day, so.
This last discussion actually was a great lead-in to Irving, my session. I'd like to accomplish the following things. One, to briefly, I think you know this, but to briefly remind you that cancer is a very complex disease. The second thing is to let you know that we are in the best of times when it comes to technology, awareness, and our ability to address it. The technologies are there, and the customer awareness and acceptance is also there. I'd like also to remind you of the famous statement that insanity is doing the same thing the same way and expecting better results. Same is even bad. Okay? And finally, because of that, to suggest to you that the Moonshot Project is about all the things we were just talking about, but doing it in a different way. Oh, I can see here. There are lots of cancers. I'm not going to go through these slides. You can look at them. But there are lots of types of cancers. There are probably about 100 different cancers, and for many cancers, there are many subtypes. So we're talking about not a single disease. We're not just talking about solid cancers and hematopoietic or blood cancers, but there are lots of subtypes around them. And this is a slide that you can look at if you get copies, which shows the incidence of several of these cancers. In this slide, I've just pulled out a few. And you can see the incidence of breast each year. We're still having a lot of people with breast cancer, particularly females. It's less prevalent in males. Prostate is where you find a lot in males. So each year, we're still having a significant number of people who are getting cancer, these various cancers, and dying from them. Cancers are, we know now, some of them are hereditary, and I'll talk a little bit about some of these, but we're familiar with colon cancer and its precursor, adenomatosis polyposis coli. We have the endocrine MEN, the multiple endocrine neoplasias, those types of cancers that give you thyroid cancers, pheochromocytomas, etc. So there are a number of these cancers that we know they're hereditary, and we have markers for them which actually lead to diagnostics. And then there are other cancers that are purely that are somatic. There are mutations, changes in the somatic cells that give you those cancers. Sometimes agents such as carcinogens, UV light, and other things that happen that we do not understand why these translocations between genes or mutations take place occur. This is a slide, I don't want to go through details of it, but it's a very famous slide, an article published by Hanahan and Bob Weinberg, Bob from MIT, which made a very important statement. This article was published in Cell in 2000. I think it was for the first time we began to realize that we're a little bit crazy to think that one particular approach is probably going to kill a cancer, because cancer cells have ability to grow, to proliferate, they have ability to metastasize, they have ways of building new cells, angiogenesis, and therefore increasing their lifespan. And they have ways of turning off the programmed cell death. Every cell is programmed ultimately to die, apoptosis. They have ways of avoiding that. And let me keep it high level. What I really want to say in this slide, though, because of that, we have different types of treatments that we have used. For example, cytotoxic, things like Taxol. We use that because we know that the telomeres, if we can stabilize them, the cells do not divide, and therefore we can stop proliferation. We can also, apoptosis, we have, this is the programmed cell death. Cancers can escape that. We have drugs which intervene there. We have drugs which affect the growth receptors, the epidermal growth factor. We have drugs which affect EGR, like Herceptin is one of the subsets of the epidermal growth factor receptor. We have others that can impact small molecules, the signals that those receptors, when stimulate, send to the nucleus to produce proliferation. Gardasil, we all know about HPV, virus-induced oncogenic activation of oncogenes. We have now a vaccine approach to treat that. So because of our understanding of the different ways a cell, when it becomes cancerous, either becomes cancerous or can avoid control either of replication or location, we have various types of drugs to address it. There are other approaches, such as nanotechnology, which is now coming into play. There are some approaches, and Irv will discuss one in detail, so I won't touch the stem cell. But we are able to engineer peptides, for example, which can target to a cell, and when they get to the cell, become internalized, affect the mitochondria, kill the mitochondria, and therefore help. So there are also additional approaches than the ones that I gave you. Now, the role of diagnostic tests. These, remember, for the hereditary, I mentioned a number of, indeed, either proteins or oncogenes or, things that we can. But what's important about these tests is that you can make decisions. A woman who has BRCA1, whose family has and has, can make a decision to have a preventive mastectomy to avoid breast cancer, for example. Or if we find a 10-year-old child has the potential to get colon cancer, we can intervene with regular endoscopies, and some even elect to have colectomy, and on and on. The whole point about the diagnostic test is that you can identify early, you can do prevention, you can have early intervention. So you can predict, you can help with prevention, early intervention. And we know, for example, if we intervene in breast cancer early, the 5-year survival is much better. Now, I only have 3 more slides, and this is the issue. NCI spends about, in 2007, spent about $5 billion to fight cancer. And only 700 of that is spent internally. So it's not that they're spending the money in Bethesda. Okay. And a lot of that money was spent on SPORES, Special Centers of Excellence, on other centers to force collaboration, but about $2.5 billion was spent on the regular individual type grants. NCI also has a very detailed, that they're building, continuing to build IT, the CA BIG program that can pull all of these together. 40 years ago, Richard Nixon declared the war on cancer, and we haven't won it yet. So, my suggestion for this moonshot program is we start doing things differently. When you understand the complexity, the different diseases, is we take a holistic approach, one that really brings all of these things together, that can focus on early identification, prevention, early intervention, and get to at least the stage where there is chronic treatment for a cancer. Second, we need a multidisciplinary team approach. It is very clear that you need the basic scientists, you need all the omics we talked about, you need the chemists, you need a synthetic biologist, you also need the engineers to be brought into this. A focus on early commercialization will also help when we do have a signal that we understand that might be helpful. We'll focus the efforts to really generate both diagnostics and therapies. So there needs to be collaboration between academia and industry. And indeed, since we know a lot also about how lifestyle changes and genetic counseling can add to that, they need to be a part of it. And the previous speaker mentioned the electronic healthcare records, et cetera. Just think of how issues around how we capture data and all of the clinical information we have that we do not use, how this can be brought together. So this is already paid for. There's $5 billion there. What if, as a moonshot project, the suggestion is NCI developed 3, let us say, regional centers, and I'm not going to prescribe, how they're run, but 3 regional centers which had these characteristics: holistic, multidisciplinary, involvement particularly of the engineers, academia, industry collaborations, and early focus on commercialization of ideas, deal with the insurance issues, etc. What if they did that and they said these centers will be selected based on their abilities to pull that sort of effort together, and they focus on a set of cancers? One last comment, recently published, I think it was in AML, in acute myelogenous leukemia. They actually did a complete genome sequence of a cancer cell from an individual and a normal cell. And they found in the cancer cell there were 100 mutations. If you've got 100 mutations and you continue to do it in this distributed way, dispersed way in which we presently study these things, think of how long it will take to find out which is the mutation that is important and what are the pathways, etc., and therefore the potential areas for intervention to find a therapy. That's my proposal.
So I'm Irv Weissman. I'm head of the Institute for Stem Cell Biology and Regenerative Medicine at Stanford, and I'm also just stepped down as head of the Cancer Center so that I could actually do something instead of administer. So I want to give you an idea about stem cell biology and how it applies to cancer and how it really is leading us to a new way of thinking about it. So every one of us is turning over cells in our body, and we must regenerate those cells from precursors we call stem cells. The one characteristic of a stem cell is that when it divides, it makes 2 daughter cells, one of which is a stem cell and the other isn't. The other one that isn't can divide a lot, but it never really makes itself. And from the beginning of the tissue to the end, there may be 30 cell divisions, but only one cell is regenerating itself every time, and that's a stem cell. We have stem cells in our skeletal muscle, in our blood, in our bone, in our cartilage. In fact, in almost every tissue, including our brains. Every one of us, no matter how old we are making, this moment, 30,000 new brain cells from the hippocampus and the lateral ventricle. That's as an aside. We developed a general method in my lab 20-some years ago to isolate stem cells prospectively, first in mice and then in humans. And the paradigm goes this way. You study the mouse first, you test the cells in a mouse, then you take a mouse that has no immune system and you put in human organs, and then you test human candidate cells in the human organs in a mouse instead of treating medical students. And then finally, you go to the FDA and you say, here's our results, and now we want to do human to human. That's a paradigm that goes over and over and over again. That's not the paradigm of looking for new cancer drugs. New cancer drugs are usually looked at in cell lines, not primary cancers as they exist. And the question is, can I shrink the cancer by 90 or 99%? Can I kill off those cells? And then they'll try a cancer that's been transplanted many, many times, mouse to mouse, and eventually they have to face the clinical trial. So in order to have a new way of thinking about cancer and cancer therapies, we had to move to what stem cells are. Now, I guess this will do that. So we studied the blood-forming system, and 20 years ago, we isolated first the mouse blood-forming stem cell, 1 in 15,000 cells in the blood-forming organ. So it's really a needle in the haystack. And then later, over the next 15 years, we isolated every cell in between, from the stem cell to the blood, and the only one that self-renews is the blood-forming stem cell. Regeneration of the blood-forming system. Whether you were irradiated in Hiroshima or your doc gave you 3 drugs to try to kill the cancer in your body depends on this cell, because this is the most sensitive system in the body. You've got to regenerate this system. Now, of course, if you get a cancer of this system, if you know every cell, you can ask which one gave rise to the cancer. And I'll get back to that in a second, but I just wanted to get this to you. Now, the general method we used was a method developed in the 60s and 70s to sort cells from a little bit of blood from a mouse. We still have essentially that instrument to prospectively isolate stem cells or progenitors, or as I'll tell you, cancer stem cells. This was a huge feat of engineering coming out of the Herzenberg's lab to allow you to make monoclonal antibodies, coat one or more of the antibodies that's colored on a cell, and they use this machine to sort them into the blood-forming stem cell and the unwanted cells. And I'm using this to remind me that we formed a company called Systemics years ago to treat patients who have cancer with their own blood-forming stem cells after they received a lethal dose of combination chemotherapy to try to kill every cell in the body. It was a very successful company because it made a lot of money. It made enough money being sold to a large pharma that it was destroyed by the large pharma company. So there's a barrier right there. And it didn't happen just once, it happens almost every time. The cultures are different. I'm not going to go into this too much. I'll just say, when we gave back to 22 women with advanced breast cancer that had spread throughout their body, their own cancer-free stem cells, and we could compare it in the literature with giving back the bone marrow or the mobilized blood, which 40% of the time has metastatic or spread cancer in it. The ones that had cancer in it, 6% alive without disease in 2.5 years. I just went back over our data, the Stanford data on the same breast cancer phase 1/2 trial, 37% alive without disease 7 to 8 years. Given that data, you would have thought this would be a therapy. But we were way ahead of our time in trying to develop an industry when insurance companies, oncologists, et cetera, had their own ideas of what was going on. That is a problem in cancer therapy, because if you have colorectal cancer, pancreatic cancer, stomach cancer, or liver cancer, you'd like to be able to destroy the liver, the place that all those cancers go first before they go somewhere else. We don't have the liver stem cell. We don't have the means to do that, but we know in animal models at a company I founded, we actually do have a liver engrafting cell. So if you could find stem cells for each of the organ systems, you could use today's therapeutics to improve dramatically the care system. But that company is gone, and that idea is very hard to progress. And we can do it in the question period. Now, you can also take blood-forming cells from one person and put it into another, especially if they're siblings and you just have to overcome a minor immune response. So if that's me, that's Frank Douglas. He just got irradiated. We give him my whole blood-forming system. The immune cells in my body that were preformed attack every organ in his body, and it's called an immune reaction that puts the patient in an intensive care unit, and it makes 2.5 times the cost for the clinical care for those patients. Now, if you don't have a cancer that could be treated by the T cells that are fighting, but you have sickle cell, severe combined immunodeficiency, beta thalassemia, juvenile diabetes, lupus erythematosus, multiple sclerosis, you'd like to have stem cells without the immune reaction. And we've done this over and over again, mouse to mouse, cure the diseases by replacing the genetically defective systems with sibling ones that are healthy. We've done human to mouse, and we are not yet at the point we can do human to human. But this is just to say this is a very big field, and it is called regenerative medicine. If Frank has got my blood-forming stem cells in him, then he can get a skin graft from me or a heart graft or a heart stem cell graft because he has my immune system and he can't reject what's me without any immunosuppression. It's so amazing to me. When I first did this experiment at 18 in Great Falls, Montana, in a small lab, I went home and told my mom, that was 1958, that it's going to be just a short time and we're going to do this all the time with people. We're just going to give them a shot of stem cells and then they're going to have a skin transplant or whatever. It still doesn't happen. The science is there, but the application of the science to clinical medicine is still far behind. Now, when we applied stem cell thinking to cancers, it was a pretty obvious thing, but I do have to say it. Cancers don't come from Mars, they come from our own tissues. You know, there aren't genes bombarding us. It's our own cells that are self-renewing that can self-renew out of control. That's the problem with cancer. All of the other properties derive from that. It's cells that are poorly regulated in their self-renewal. Because most of the cancers in humans haven't gone to the ultimate stage that every single cell in the cancer is a cancer cell that transfers the cancer, we found, Mike Clark and I and Tanisha Ray and Sean Morrison, that within a cancer, about 10% of the cells are self-renewing and about 90% are the daughter cells. And that's excluding, of course, if it's a breast cancer, the new blood vessels coming in, which you talked about, the immune reaction, the inflammatory reaction, the stromal cells from the host that take care of it. But if you even look at the cancer, on the average, about 10% are self-renewing. And when we put a breast cancer stem cell that we've prospectively isolated into the breast of an immune-deficient mouse, it and only it transfers the cancer. When we take acute myelogenous leukemia, about 10% of the cells have surface markers. We know what the other cell is, that it was the stage at which it comes from. When we look at this, again, as Frank says, we have accounted up at least in each of these leukemias, at least 10 changes that made it to a leukemia. Now those 10 changes, these aren't like the sequenced mutations, but these are 10 changes that avoid programmed cell death, avoid the immune response, keep the chromosomes long instead of let them get shorter, and so on and so on and so on. Within a single cancer cell, you have a catalog of events, each of which probably occurs 1 in a million times in a single cell division. So to accumulate the 10 events takes a long, long time. And now we go back and look at our leukemia patients that are cured, and sure enough, we can find the precursor of the leukemia in the patient. And many of them, you can go 10 years, and it still won't tip back to the leukemia because it's got to go through 8, 9, or 10 events, each of which is rare. Now, those events are, of course, opportunities, because each one of those events adds to the progress of getting a cancer. Now, what people have done with genomics and gene expression analysis is to take all of the bone marrow. in a patient from leukemia, only 6 to 10% of which are the scary cells that can cause and spread the leukemia. And they look at the gene expression, and 90% of the signals they look at are irrelevant to the cancer stem cell. When we purify that leukemia stem cell, and we know what stage of development it is, and we compare it to the normal stem cell, or the normal multipotent regenerative, which is the stage, suddenly come pouring out massive numbers of repeatable genetic changes from patient to patient that were never seen before. And so, if we were pharmaceutical companies, we now have a few targets to tell them that are small molecules that might do this, but I want to remind you, well, I won't remind you, I'll just tell you. When you have a cancer, you have 10 to the 11th to 10 to the 12th cancer cells in your body. That's when you first find out that you got a cancer. If 10% of them are cancer stem cells, so it's 10 to the 9th to 10 to the 10th. If you get a drug that kills 6 logs of the normal cells, you've shrunk that tumor down tremendously, but if you didn't kill the last cancer stem cell, it regrows. If this cell spreads into the blood, it doesn't matter. If this tumor stem cell spreads to the blood, you're in trouble. So now we're talking about, quote, rare cells, which you have to purify ahead of time, and then you get the information for diagnostics. So if I were at Siemens, I would say, for example, can I have nanoparticles that target that cell? Well, we have a collaboration through the Siebel Stem Cell Foundation, by the way. He's left us. He doesn't even want to hear this. But, okay. Hey, Tom. We have a Zurich nanoparticle guy who's coming to Berkeley, coming over to Stanford to work with our engineers. And this is the point I want to tell you, that we have this unique opportunity that we've come upon engineering problems. So we need to have a cell sorter that doesn't look at one cell at a time to pull it out. We need one that's massively parallel. And a lot of people at my company say, oh, you couldn't do it, or they put a few people on it, not enough. But a Japanese scientist went to Sony and they came up with a prototype with microfluidics that sorts cells, 2,000 cells per chip, plastic chip, and you can get hundreds of them to do it all in parallel. So it's not an impossible engineering problem. It's one that needs an economics to drive it. And certainly Sony isn't going to do that because they have a big bottom line. The bioengineers, Steve Quake, for example, at Stanford, can do all of the biochemistry that usually it takes 1 billion cells to do, can do it with 1 to 100 cells. So we actually can look at the proteins, the RNA messages, the changes in the RNA messages, and start to come up with things. So this was the field that you mentioned, microfluidics, and it is going like mad. So if you're investing in academic centers, make sure they have a terrific engineering department, terrific physics department, terrific imaging, as well as the cancer cell biologists and stem cell biologists. I'm going to just end up by showing you one example, because we looked at human acute myelogenous leukemia. We found a molecule that's always overexpressed in the leukemia, whether it's a mouse leukemia or a human acute myelogenous leukemia or the late stage of chronic myelogenous leukemia, and now so far every solid cancer we've looked at. By the expression of one molecule, the cancer cell says, "Don't eat me." That's got a normal physiological function I won't go into, but that means cells that professionally eat can't eat it. And what happens if you block the "don't eat me" signal? Single antibody treatment in mice that have a human primary acute myelogenous leukemia, transplanted them 2 months ago. They're about to die. We give an antibody treatment. The blood cells goes away, and you end up with a bone marrow that's almost right. So the final point I'll make is, any single therapy like this antibody therapy will kill off most of the cells except 1 in a million will already have a mutation before you started. That's a loss of that particular molecule. Any company that treats with a single monoclonal antibody is not going with curative intent. It has a financial intent. It makes the disease more chronic, but the disease comes back from the pre-existing variants. We've known since the early 50s that you need 3 independent targets with 3 independent approaches to go for curative intent, whether it's a bacterial infection or cancer. So we have to get a whole new point of view from people in this field, and I'll just say that thanks to Proposition 71, the California Institute of Regenerative Medicine is funding regenerative medicine and cancer stem cell work up to the point that maybe the venture capitalists will talk to us again. Thank you.
All right, good. Well, I don't want to get in the way of lunch, but I do want to just be sure we address a couple things. I know we have a question from the audience, so I don't want to dally for that. Can we get it on?
Is it on?
Yeah.
Okay. So the healthcare session of the discussion leaves one kind of obvious point open, which is why is preventive care not paid for by the insurance companies? And this has been on the table for a long time. It's kind of mind-boggling that a simple, well-understood problem is not solved yet. Can you please talk to that, the experts?
I think the problem is that you don't necessarily stay with the one insurance company. So one insurance company pays to pay the protection, then you can move to another insurance company. So you're not paying for a client that you stay forever. And so that's why the insurance companies tend to be reluctant to pay for for preventative medicine.
No, I think that is indeed one of the major problems, is that one insurance company pays and then another insurance company, when you switch, either because you've moved or whatever the reasons are, let's say you change your job and that other firm has a different company, then that company gets the benefit. And so that's one of the reasons. But one of the things I wanted to say in response to your question is there is an opportunity here. Because in Europe, in some of the countries of Europe, where the government bears a fair large of the insurance, healthcare, insures the population, there is really an opportunity for the preventive part to be approached. And I do not know why they do not do it. I'm responding to your question, that if you want to... No, I appreciate it, but I am responding to your question. Well, I can answer that part of it, is that in a real sense, our present model isn't working. Okay, so, whether nationalization is the answer, I'm not so sure. There are downsides to that, we know. But in terms of doing the experiment, and to get around the problem that Reg mentioned in terms of moving from insurance company to insurance company, Europe is one place, or some countries in Europe, where that experiment can be done and where indeed one can collect the data. There's some of the Scandinavian countries, for example, that really have a very robust healthcare system and where they track people, etc. You can do basically the healthcare economics of it.
I think there's also a very important function in the European health insurance systems that is now on the political agenda in the United States, which is to have a cost-effectiveness analysis organization. In the UK, this is called NICE, the National Institute for Clinical Effectiveness. The problem is for each individual health insurer here to make their own decision about whether something is cost-effective as a preventive approach is very difficult. So you see most of the health plans or insurers covering vaccines, for example, because CDC plays a role in determining when a vaccine is really recommended for a population. But for many of the things we're talking about, they go through a long period of being controversial, then a period where some plans pay for them and other plans don't. And in that period, these are still quite expensive interventions usually. And so it's a very difficult decision. So I think that this is one area where we talked about energy and other areas earlier today, where national policy can play a very important role in growing the opportunity for inventions that can really make people healthier.
Another question from the audience here.
I'll talk.
So the previous question highlights that the United States, and Paul Camuti's talk highlight that the United States healthcare incentives are terribly aligned for all kinds of useful innovation. The title of the conference is Global Technology, and I'm wondering whether it makes a lot more sense to invent cost-effective and then maybe later more personalized healthcare technologies for the middle of the pyramid in emerging markets, and for Europe, where there's a more rational alignment of incentives. I mean, the United States system is broken, so it's expensive, and people are going to be sicker and deader as a result. And the solutions for that probably aren't primarily technological, and this isn't the place to debate that. But as a practical entrepreneur in healthcare technology, that's where I'm aiming innovation that relates to lowering cost. And I'm wanted to provoke you guys to discuss the plausibility of that. Most of the discussion was all about United States healthcare, and it was as if there's not going to be most of the world's diabetics in India and China in 15 years. In India, most of the world's diabetics are going to be in places like that. So how about inventing against that?
Well, I'm a European, so I can comment on it. But you see, the problem is so strange that we have a combination of problems in the United States and Europe, and each one of them is leading to a worse healthcare. In Europe, what happens is that there is no incentive of acting economically because it's the governments who are paying. And the notion of efficiency in governments, we know where it is, right? It's almost zero. Now, in terms of innovation, when you go and have a discussion with a public system, what happens is that they barely survive on what they are doing now. So there is no money to invest. And so you have a problem on Europe that is public, so you can do a lot more because there is only one guy who pays for it, so you would assume that that's ideal to introduce this innovation we are talking about, but there is no incentive to do so because there is no economics for it, and two, they have no money to invest.
I mean, you set a bounty on, you have a prize for a cost-saving invention that's going to be, whose benefits will be shared over a whole region. That's plausible. That's using a prize incentive. It's the sort of thing Tom Siebel was talking about earlier. It's very rational. It's because a lot of these things are about externalities and social group things across an insurance pool. So that's what prizes and a big pile of money is for. I mean, the US military developed antibiotics during World War II. They found the money for it, and it saved the country.
So I think Molly has a response.
Well, I just wanted to give an example, and again, it doesn't come from biotechnology, but from IT and devices, but depression is one of the most expensive conditions in the population in Europe, the US, all around the world, and we spend a lot on pharmaceuticals for it. But in the UK, because they have this national approval system with NICE, they approved cognitive behavioral therapy, which uses a computer for the individual patient to manage their depression. And it's accompanied by pharmaceuticals, but reduces the need for pharmaceuticals and really improves their function. In the United States, this is only being adopted in the Veterans Administration, because the Veterans Administration is like the UK. it is on the hook for the cost long term. And so this is precisely why I don't myself believe that the answer is to have Medicare for all, because Medicare doesn't do this. The answer is to have a national system or entity that at least looks at the cost effectiveness, and that's why I was excited by what Reg had proposed.
I think that.
One question. If the Obama administration came to you and asked for 3 or 4 top points of what you recommend as the suggested change in the healthcare policy, what is your recommendation?
Sounds like a good lunch topic.
Yeah, that's a 3 or 4 from 20 people is about 40 answers. So maybe we should talk about that over lunch. But I do think we are in for some change in terms of health policy. So I think that's around the corner. I'll turn it back over to you.
All right, thanks. So, one thing I'm really glad is that was a very heated conversation, and that's actually what we want. So that was wonderful. What's going to happen now is we're basically going to have lunch. I believe that it's back here. I probably need some help to know that that's for sure. But people will go back and get lunch and basically bring it back. We'll give you a little time to get situated, and then we'll have our keynote address, just after everybody starts eating. And just give yourself a little time, and then we'll start that. In the meantime, I basically don't leave till you fill out this. Don't get up till you fill out this form. Very important to us.